welcome to STR IAS. So in the previous lecture, we were seeing about cell organelles. So one of the cell organelles or two of the cell organelles which we have seen is cell membrane we have seen and cell wall we have seen, and we have seen that cell wall is only present in plants but not in animals. So in this lecture, and we have started about mitochondria. So mitochondria is also called as powerhouse of the cell. Then mitochondria does something called aerobic respiration. Every cell will be having mitochondria. So whatever the oxygen we inhale, it will be going to every cell at cellular level. It will be it will be doing something with glucose. It will be breaking down glucose and it will be producing energy in the form of adenosine triphosphate, nothing but ATP. And for every function we need some energy. That energy will be provided by mitochondria, and that's why mitochondria is also called as powerhouse of the cell. And mitochondria will be having a single circular DNA molecule. So other than nucleus, which is the cell organelle which is having DNA or genetic information, is called mitochondria, right? So then we should see about the structure of mitochondria. So mitochondria is double membrane structure. Inner membrane and outer membrane will be there, and these finger shaped structures are called cristae, and there will be matrix, nothing but fluid in that, and there will be some DNA in it, right? Then we should see about functions of mitochondria. So mitochondria, the major function is like powerhouse, right? It should produce some energy in the form of ATP. That's all we should remember. And also it will be storing calcium, and also it will be programming the cell death. So what is the word for cell death? Apoptosis, nothing but death of the cell. Just remember the word, just because we can see in the PYQs this word, right? And also it also produces heat through a process called thermogenesis. Thermo means heat, genesis means taking birth of thing. So heat will be generated in mitochondria by mitochondria. And also it also performs metabolic functions. So all our metabolism rate in the body depends upon how our mitochondria is functioning, right? And also it will be signaling a key role by signaling hormones, and it also will be doing intercellular signaling. It will be like, for example, our pituitary gland. If you want your testosterone or progesterone or estrogen to stimulate, who will be signaling those cells? So intercell signaling will be done by this mitochondria, right? So we'll be seeing more detail about mitochondria in human reproduction in the upcoming lectures. So the next cell organelle which we'll be seeing about is chloroplast. So chloroplast, it is the organelle which is present only in plant cells. So we have seen that cell wall and chloroplast are the things which are present only in plant cell. They won't be present in animal cell. We should not expect these things. So chloroplast only present in plants, and it also contains a pigment called chlorophyll, right? And also it performs a functions called photosynthesis, with which it makes food for plants. So all of you might have studied in your class 10 or class 9 about photosynthesis, Krebs cycle and all. So this chloroplast also will be having some small circular DNA, you should remember. So in plants we'll be having DNA in nucleus, and also in mitochondria, and also in chloroplast will be having DNA. But in animals, only in nucleus and mitochondria will be having DNA, right? Then we'll be getting into something called nucleus, the next cell organelle. It is also called as brain of the cell. Why it is called brain of the cell? Because all the information regarding all the functions to be performed, which function to be performed when, all these functions will be coded in as an info in something called nucleus in the format of genetic material. So we'll be looking at genetic material in more detail, and in the upcoming lectures also we'll be describing more about DNA, RNA, how it forms proteins or enzymes. So you can see that this is the shape of DNA, and DNA will be nucleus. So nucleus will be having nucleus pores for the material to allow inside and outside. So RNA should come inside and RNA should go outside. So for that we have nuclear pores, and we have genetic material called chromatin in this. chromatin. Why chromatin, sir? Why not DNA directly? We'll be looking into it. And we have at the center of the nucleus, we have nucleolus, and this gap, the membrane which is like gap, it is called nuclear membrane. And we have a fluid also in this fluid. This is also called as nucleoplasm, right? So this fluid is also called as nucleoplasm. Then next, so we can see that this nuclear membrane, this only will be there in eukaryotes. In prokaryotes, there won't be any nuclear membrane, because uh, in prokaryotes the cell is not fully developed, right? Then, so what is the function of nucleolus, which is present in the middle of the, so we can see that this is a nuclear envelope, nothing but nuclear membrane. We'll be having something called a nucleolus, chromatin, so, and fluid, nuclear matrix, nothing but nuclear fluid in which all these things are floating, is called nucleolus. And what the function it will be doing? It will be doing RNA synthesis, rRNA. So we'll be looking at what is rRNA, what is mRNA, what is tRNA and all. And we have chromatin, which is a condensed form of DNA. So we'll be looking at this, like what is this condensed form and all. So I told that cell will be having some genetic material. So genetic material is nothing but DNA. Genetic material is nothing but DNA, right? Genetic material will be in the form of DNA or RNA. So what are the characteristics of genetic material? It can replicate. It can replicate, and also it should be stable, and there should be scope of mutation. There should be scope for changing, right? And they should express Mendelian characters. All about this we'll be looking in the next lecture. Okay, so what is it? Change should happen. Why it should be stable in the sense the structure should be stable. So we can see that DNA will be having double helical structure. So this double helical structure should be stable, but it should change. It should be having scope of mutation. In that also, like, composition should change at some point of time to get more good characteristics or bad characteristics. Whatever happens, it happens due to mutation, and it should express Mendelian characteristics, nothing but law of inheritance. For example, a baby will be getting 70% of intelligence from mother and father, right? So this intelligence will be coded, or this information will be available in gene. So that gene should be sent to these daughter cells or to the babies, right? That is called laws of inheritance. So these are the features of genetic material. And the first genetic material which we'll be looking at is DNA. So what is the full form of DNA? Deoxyribonucleic acid. Deoxyribonucleic acid. So what is the structure of DNA? Double helical structure. What is the structure of DNA? Double helical structure. And what do you mean by deoxy? No oxygen. Ribose. So the no oxygen will be having sugars and also phosphate backbone for rigidity or stability. Then nucleic acid, it will be joined with the help of something called nucleotides. So too much of chemistry is here, so no need to remember all these things, but just remember nucleotides means nitrogen bases. We'll be having two types of nitrogen bases. One type is called purines and the other type is called pyrimidines. So purines are adenine and guanine. Okay, just remember with the help of pug. In DNA we'll be having nucleotides, nothing but nitrogen bases. There are two types. One is purine and another one is pyrimidine. So in purine we have adenine and guanine, and in pyrimidine we have cytosine and thymine. The bond between adenine and thymine will be double bond. The bond between guanine and cytosine will be triple bond, right? So we can remember this with the help of at the girls club, right? A and T will be together always with the help of double bond. G and C will be always together with the help of triple bond. A and G are purines. T and C are pyrimidines. What are these purides and pyrimidines? These things are nothing but nitrogen pairs or nitrogen bases in DNA. So just look at the structure of DNA now, right? So DNA structure, we can see that so these are the, so you can see this, right? So these are the nitrogen bases which will be holding this two helicals structure, and this is a sugar phosphate, so sugar phosphate backbone which will be holding. So we'll be having like this nitrogen pairs, and they will be holded by sugar phosphate backbone which will be giving stability, right? And these things are called base pairs. Fine. Then so there is a diagram directly from NCERT. So we can see that phosphate, and we can see these nitrogen pairs here: adenine, thymine, guanine, cytosine. These are like connected through hydrogen bonds. We can see double bond here, we can see double bond here. Between G and C there will be triple bond, and these are like rigidity is given by this sugar phosphate bond. So this structure is of sugars, right? Then so no need to remember all this structure and all, no one will be asking you. Just remember that there will be base pairs made up of nitrogen base pairs in the between, and they will be connected to each other with the help of this. They will be giving stability, or one structure will be given by sugar phosphates bonds, right? What are these nitrogen pairs? ATCG. A and T will be connected by double bond. G and C or C or G will be connected by triple bond. Then I have told that DNA, but sir, you are not telling DNA directly, but you are telling that there will be chromatin or chromosomes in nucleus. Why? Because if you see DNA molecule like this, it will be there all the information regarding our eye, heart, brain, kidneys, lungs, how they should function, hands, nerves and all. So the thing is, if you take all this DNA, it will be of 6 feet in each cell. All the information in each cell, it will be 6 feet. So in that scenario, can we fit a 6 feet substance in a very micro level cell? So it doesn't happen, right? So for that we need to condense it, we need to compress it. So it's a very long molecule and it needs to be compressed to 2.2 meters. So it needs coiling to fit and into cells, right? So DNA coiling should happen. No need to worry, all these things they won't be asking, but we'll be looking into it in detail. So DNA is very lengthy, 6 feet it will be there. It should be condensed, so it should be condensed into chromatin. It means that I'm coiling it, I'm coiling it like this, and I'm fitting into nucleus of the cell. So for that I need something called histone proteins. Histone proteins are nothing but there will be some proteins like this. I'll be binding them on this, right? I'll be coiling them on this. I won't be coiling, there are some enzymes which will be coiling them, right? So what are the proteins which will be giving stability, or while condensing or while compressing, what are the proteins used? Histone proteins are used to attach to DNA, and DNA will be attaching to that histone protein, and they'll be getting coiled as chromatin. Alright, so histone protein, like there will be many histone proteins. the DNA will be coming here, it will be attaching, like this, it will be attaching like this, then there will be one more histone protein, it will be attaching like this, right? So all these things, so each thing where to the histone where DNA is getting coiled is called nucleosome. So nucleosome, many nucleosomes will be getting fitted into one nucleus, right? It is called chromatin. So 6 feet length wire, double helical wire, we can make it into 30 nanometers, and it is fitted into a nucleus of the cell. So look at how complex our life is. So what are the functions of DNA? So it stores genetic information. So the information from our mother and father, their genes, it will be stored. It will be found in all cells. So every cell will be having genetic material. Without genetic material, they won't be knowing what to perform, right? And it helps in formation of mRNA. Why do we need mRNA? Why do we need protein? We'll be looking at